always love the beginnings of these kinds of conversations where we all get started and dig in together. And so then it's always a little bit awkward to restart, but we can restart, but please let's keep this open and conversational, because that's the goal here, to be all learning together. So this is our third engagement meeting with the GRIN Therapeutics and the GRIN community. Just to introduce myself, I'm Hilary Savoie. I am the head of community engagement and communications at GRIN Therapeutics. And for the last 2 weeks, we've been kind of coming together and having these conversations, getting to know each other, and setting some priorities for how we work together. And today we're going to keep moving along in that discussion. Our goals today are really to talk about clinical trials, share our experiences with clinical trials, and the process of development of new medications, and open a transparent dialogue for how we can best work together to bring meaningful medicines forward for the GRIN community. A couple housekeeping items. We are recording, as everyone knows, so please, you're invited to come on camera, join us, talk, and show up in whatever way is most comfortable for you. We'll have a Q&A at the end of the session, sort of more formally, that will be led by Liz and Keith, but feel free to ask your questions as we go along, interrupt, type in the chat, however works best for you. And with that, I think we can move on to just stepping through the agenda for today a little bit. We're going to talk about what a clinical trial is, how they look a little bit different in rare indications, what it's like to participate in a clinical trial, where, when, and how the community can provide input and be involved. I'm going to give some examples from the community that I have worked in as an advocate in, and from Luke as well from his work. And then we're going to talk about, further opportunities to engage. Some intros, we've all been talking already. Pierre Andrea is here today, our founder. Hans, our CBO. I don't think Bruce has joined us, but he may later. And as I said, I'm here, I'm Hilary. And we have Luke here, Luke Rosen is the founder and board chair of KIF1A.org. And he's also the senior VP of Accelerated Development and Community Engagement at Ovid. Luke is a real pioneer in bringing community and patient voices into the drug development process. And he will be here today providing his endless insights to this whole process. So we're so grateful that he's here. And with that, I think just to give us a bit of background, I'll hand over to Hans and Pierre Andrea to just walk us through what kind of the traditional approach to clinical trials is, and how that looks a little bit different in a rare disease indication, and why those approaches have to be different. So with that, Hans, Pierre Andrea, you want to take over?
Sounds good. You want to start, Pierre Andrea?
Okay, I'll get it started. So you see, it's a very informal way. We haven't set up a clear script, so let's keep it a very conversation as a dialogue. I guess, reflection, someone was saying about COVID, maybe for people that haven't seen clinical trials, they probably learn a bit more over the last years with all this COVID vaccine and clinical trials, but nevertheless, I think I'm going to repeat some of the stuff that might be known by now to most of you. So this is like, as you progress, as you come up with a molecule that has promises, you have to go through all these steps to get approval. And the reason is that you need to learn and bits and pieces of all the molecular characteristics, starting with safety and then eventually with efficacy, and try to do it in a way that it doesn't, there is always a positive benefit risk assessment of the overall situation. So you expose gradually more and more patients as you learn. And you start with phase 1, that as you see here is healthy volunteers, and then you move into efficacy in a smaller group of patients, and eventually larger patients sets. And by traditionally the FDA says that it's going to approve a drug if you have 2 confirmatory studies, well designed, that shows the efficacy is there, of course, with safety and tolerability. That's kind of the basic for everything is always done. And then they come later in the next slide the exception. But before we go to the next slide, I'll pause here to see if Hans want to add anything, and on there are questions. Maybe just to say that traditionally this process might take up to 10 years from initial phase 1 to the end of phase 3. And then normally if you count all the failures, that can cost over $1 billion to the industry to put up all this process. That is not only the clinical, but everything goes together. So it's very lengthy, very laborious, intense, and requires a lot of people working in all the concept. So I'll stop here.
Yeah, and I would just see if there are questions from the folks in the Zoom call. But you know, to Pierre-Andrea's point, it's really based on the scientific methodology, based on large patient population statistics, and you know, it's very well regimented in what the FDA, or in Europe the EMA, expects as you take a program through the clinic. And what we'll turn to next is more about, that's not the situation that we're in now, which creates opportunities and challenges, and we'll talk through that. But you know, is it clear? Are there any discussion points or things that we should address before we get into that? If maybe we keep going.
Well, one thing, sorry, can you still go back? Because I would say just, I don't want to make things too complex, but there is even here a simplification, you know, between phase 2 and phase 3. Sometimes we have a phase 2B, that there are normally different doses of a drug that they are tested before you go phase 3. That is a confirmatory study. And also between phase 1 and phase 2, sometimes there is a phase 1B where you do the safety and tolerability in actual patient population, not on healthy volunteers. That's kind of the orthodox way to distinguish studies. Then you also will hear other time use other language like proof of concept. What is that proof of concept? And you know, when you prove that the concept is there, and meaning that normally that you have the concept is that you want to increase the efficacy, well, have efficacy with the compound. So the proof of concept is normally the first time you study the efficacy in the specific target population. So you might see, I mean, this is similar to the discussion before. Mutation genetic variants is of phase 2 is sometimes equal to proof of concept, according to most of the people in the field.
Yeah,
So should we move to the next?
Unless there are questions, we should. So as I was saying before, this is like the standard, you know, 2 confirmatory well-designed studies. The FDA demands to get approval. And if you have an orphan condition, they say this is the same for an orphan condition. But having said that, they normally are more prone to accept exceptions. I can say that around between 40 and 50% of clinical studies for orphan indications in neurology have been approved with a single study out of the single phase 3, what we call, you know, pivotal studies ourselves. So pivotal means that is material that you submit to the agency as a major evidence of efficacy, and of course safety tolerability, but the primary endpoint is efficacy. As I said, you know, almost half of the neuro-orphan studies have been approved as a single study. So that means that there is an acceptance to deviate from the standard process of all these stage phase studies. And that means that the overall development plan can be compressed in timelines. We are not looking anymore of 10 years from phase 1, start of phase 1 to approval, shorter timelines, but also a smaller number of patients, because that's inherent in the disease, that there are not enough subjects to run comprehensive studies. So they accept that the evidence, in view of the limited number of patients, but also in view of the severity of the disease and the lack of approved efficacious treatment, there is an acceptance of smaller studies and shorter programs. As going back to the example of COVID vaccine, you saw because of the highest need, you know, that there were compressed timelines and acceptance of deviate from what is an orthodox development program. So there rules, but also exception, and orphan disease clearly represent an opening for making exception. And with that, so we have to go more on specific cases, we have a phase 1, phase 2 that can be combined because maybe the safety and tolerability has not been established yet in the specific patient population. And so you can combine two objectives. You want to test the safety and tolerability on the specific population. And in fact, you have a primary measures, a primary endpoint, safety and tolerability. But at the same time, you want to learn also about efficacy in a way that is sometimes not confirmatory because it's smaller sample size, and also because it's initial stage, and also because you need to understand first what are the dose range that you should be able to test. So there are a lot of learnings to gather in this early stage about the specific patient population, and this is very, very, very often what happened in the indications that they're similar to the one that we are interested in. Any question about this point?
I would say that whenever you're going into human subjects, you've got a very strong safety rationale. You've got a strong rationale for why the medicine should work. And in the case that we're talking about, we do have experience in the patient populations. So it's just confirming the experience as we go. So just to balance that, this construct is really understanding what's important about what we know about the medicine and the impact we expect to see, and working together, not just with the FDA to come up with a process that is going to help us to address these really important questions in a different construct and a different timeline and a different number of subjects. So it's working with the company, with the FDA or the regulatory bodies, but also with the communities and the families, as we've talked about in the last couple of weeks, because when you are trying to measure what's important, there are some things that are standard and known in the industry, like certain seizure readouts, for example. And those tend to get read out because they're things that people understand and can quantify. But there are other things too, right? And understanding what those elements are and how you measure it, that's really important. And so it's a dialogue because you're trying to optimize this clinical development program that we showed on the other slide to something that looks a little bit different and looks more like this. It's really that ecosystem that kind of is coming together to figure out how do we give this the best chance of success, and how do we understand what's really important and how to measure it. You do have medicines that fail because you're not measuring the right thing, but actually could have been good medicines, right? So that's kind of one thing. The open label is also an important concept in how that fits into the trial, but that's usually always something that you'll see in this kind of population that we're talking about as well. So we have that as a different axis here underneath. And so that I'm just trying to focus the conversation on this piece. The next slide we'll talk about what does it all mean, from an experience perspective, be a part of this. But I just want to highlight some of the nuances here to add to what Pierre-Andrea said. So maybe I'll pause there.
I think.
Go ahead.
Sorry, Hillary, go ahead.
Oh, I was just going to ask Hans to explain a little bit more about what an open label extension is, because the community might not be super familiar with that.
Yeah, so, and this can go in a couple of different directions. So I'll start it off and maybe Pierre-Andrea can fix what I say. But, in the strict definition of an open label extension, I guess you would say that the trial, if we take the phase 1/2 at the top here, the left, you've got a certain time window in which you're measuring and monitoring how the medicine is interacting with the subject, right? So maybe you've got a 12-week study or what have you, where you're measuring the measures that we've set out to measure, right? But it's an ability to continue with an open label extension. So the kiddo or the patient can continue to take the medicine, and you continue to watch and monitor how the medicine is performing over time. That's kind of one context and how it's written now. So, when the trial needs to be short to measure, or the appropriate length to measure the outcomes that we're hoping to measure to demonstrate that it's working. But that doesn't mean that it stops there, right? There's the open label extension. You can roll over or continue the trial after the trial ends, and you measure whatever the elements are from that study. But it's an opportunity for the patients to continue to take the medicine, and us all together to continue to learn how that's improving over time, or how things are tracking over time as the patient continues to stay on the medicine. So was that clear enough?
Also,
Yeah.
I think important to add on to that is that if it's a placebo study that is coming to an end, an open label extension allows once everything is unblinded, allows the families and patients who are on a placebo arm to roll over and be guaranteed to be on drug, which is part of the open label extension too.
Yeah, so there are two major objectives. One is that if a patient is receiving a benefit for what is listed there, phase 1-2, we want to ensure that its benefits continue to be there, because we don't want to withdraw the drug. So that's first, to ensure that. And second, we can learn more, what happened in the longer term, that we can always observe in a study that is reducing time. And the reason it's reducing time is because if you do, ideally, you want to study the drug as long as possible, but then things happen and the patient might have a flu, have something, they cannot stay on the trial, and then you start missing patients. Then you ended up with a long study with very few patients that have completed. So we tend to try to optimize that to a time frame where we can get meaningful information and ensure that the patient can follow certain rules. And it's not patient, the entire family will be involved. And so that's also just give a reason why there's a time bound of a certain weeks or sometime month. On that open label extension, there are 3 words. Extension, you learn now, that is an extension of a previous study. Open label means that everybody knows what's happening here. The parents, the doctor, the nurse, whoever is involved knows that the kid is taking a medicine, a drug, an active component. The other alternative is that you do a placebo control randomized study that we normally call double blind. This is a lot of language and nomenclature, but double blind means that both the doctor that gives the drug as the parents and the kids, of course, they are unaware if they're taking an active drug or placebo. So then we call it randomized, because someone get randomized to placebo group or the active group in a random fashion. Nobody knows which group they belong. And so that whatever happened is not biased by the information of knowing that someone is taking the active or placebo. So that's why we call it open label, because it's the opposite of being randomized control trial.
And I see, Jillian, do you have a question regarding that?
Yeah, I was wondering a little bit. First of all, I'm not a doctor, I'm just a parent, but we've just kind of completed a trial for our daughter with L-serine. Our daughter is loss of function. So, and when she started taking the L-serine, we were told that we would see the first effects within 6 to 8 weeks. Well, actually, it took us about 3 months to see the first effects, and it took about 6 months before she was really gearing up, coming to speed, and we really saw the improvements. And in the end, it proved very worthwhile for our daughter. She actually improved her IQ with 10 points, so it really worked. But if I hear Pierre-Andrea say things like, we want to do a trial of 12 weeks, and of course, I don't know how that works for Radiprodil, but in my personal experience for our children, they can take a longer period than expected or than perhaps normal to gear up and to see the first real effects of a drug.
Yeah, I
So
hear you.
I'm wondering, because then you're already in the access phase of the trial, in the you continue to take the drug, but the actual period for which the trial was intended is then already over. How do I have to see that or interpret that?
Yeah, it's a problem that we always face, and the solution. Well, let me say first that when we do early development studies, we never do an optimal, ideal world scenario. We always have to compromise, and unfortunately, for a number of reasons, on feasibility, on being able to get to the finishing line, and there is a discussion or at the individual level, of a group level. It's a very complex matter, and we have, unfortunately, to compromise. When you do a compromise, we can never do everything you wanted or you're supposed to be doing. And one of such things is the duration, where there is one caveat, as I said, keeping it, on a patient for a long time sometimes is a bit more complex, and you risk to lose the benefit of a group because you ended up with no group. Second, when you do first time on a patient, there is the safety and experience that you have with the drug that you wanted to keep it as a time where you have a checkpoint. And this is like after first trial, you say, okay, everything is going well and there's no issues, and the patient is actually seems to have a benefit. You don't have a benefit that is ultimately, clear cut. And you say, okay, the benefit risk is positive. So there seems to be a perceived more benefit than anticipated risk. We go into longer, that's why we have an open label extension that we allow us to observe a patient that is actually has potential to benefit more than anything else, to continue with the drug. So the effect that you saw is actually can emerge. So again, a compromise, because all the patient will be in that situation, but that's is to try to keep more on the longer term in a sense. And of many exceptions that the agency will do, looking at this condition, is to say, okay, I understand this is no control, it's an open label, but you have all these patients and all the parents actually telling us that this is what's happening, and it's important evidence, of course. And I must say here, because that might be a question later, the evidence, the way it's weighted, is very different if it's in open label and on a control setting. So the value is like, someone, coming out of a blind study where you don't know what is treated active and not active, has a lot of weight. An open label has much less weight in front of the agency, in front of everybody else looking at the data.
I think that this is why it's so important to engage in these kinds of conversations, because part of what our job is in designing the study is to make sure that we are looking at things that are measurable in the time period that we have to work with, and that we're looking at the appropriate measures. And as parents, you are experts in helping us understand what might be reasonable to measure and how. So that's such a good question, and I think a big part of why we need to have all of these conversations and many, many more going forward, so that we understand the best endpoints to be looking at.
If I could add to that, this is such a great and important conversation, because at the end of the day, I really think that it's all about endpoints, outcome measures, and criteria, right? And if you develop that with the community, then you're going to have an accelerated process. If you develop that without the community, you're going to have inappropriate endpoints that don't quite represent the disease and what a meaningful change would be. You're not going to have there's that saying that is so true, that if you can't measure it, you can't fix it, right? And so there's got to be a way to turn what many people call anecdotal narrative, which is, hey, my kid is doing better, she's walking differently. There's got to be, the trick is to somehow quantify and translate that anecdotal narrative into a standardized measure that FDA and other regulatory agencies accept and have seen before, right? And I think it starts with early regulatory engagement, right? And so if a company can facilitate in an appropriate way an early engagement with the regulatory agencies, then the regulatory agencies are going to know from day one, this is what's meaningful to the community. And then the company and Hillary and everybody can work with, and I learned a lot about this. Hans and I have worked in rare epilepsy clinical trials in the past, and learned a lot about what doesn't get done right and what needs to be done differently. And I think it's quite clear that what needs to be done differently is having that community integration into the development of endpoints that are, both, there's that statistically and clinically meaningful, and then there's what's really meaningful. And when those two kind of merge, you have what I think could be a successful clinical trial.
Yeah, no, look, thanks for saying that. I was going to say the same. I mean, there is a, the point here is that the agency, as all of us, we don't know really how to manage this, these difficult problems. And so they're very receptive and they're very collaborative, but they need to have the right input. And because, we need to explain to them what are the problems in a way that is the most accurate, and that so that we can design the study altogether in a way that is going to capture the essence. I think this is very important. I guess, Hillary, I'm really keen that we go to that experience that it was done before in this space.
Yeah. Because
of the acceptance. If it was 10, 15 years ago, we will have a probably different conversation. But now the agency has kind of open, really the communication. They want to hear, they want to learn from the community, and how to best do this is very important. Absolutely.
I can give you an example too, Hillary, whenever you want, about something, part of a study that I worked on before where that was critical in change and saving at least 2 years and probably millions of dollars, right?
We have
just let me know.
Why don't we move through a few more slides and then we can get to that story, because it's a regulatory involvement story.
It's about how, if we didn't have families in the room when we were coming up with a protocol, it's a very tangible example of how things would have gone down the tubes. So.
Okay, well, why don't we just take a peek? We can move through a couple more slides and then we're going to come back to that story, because I think it's really important that the families here all understand the kinds of impact that they can have. But I just want to wrap up kind of the notion of what a trial is. And, I think we all understand that clinical trials are very complex to be a part of. And I think it's important to just acknowledge that, communities get really excited about them. And then when they think about what is actually involved, it is a bit daunting. So I just sort of put together a few, acknowledgements of, there's increased hospital stays and site visits and testing, and there's disruption of routine, and all of that is very true. But on the other hand, you also get to learn more about your child's condition and be in the process of contributing to science and helping the community. But these are all things that we are also weighing on our end as we look toward trying to ease this process for families that are involved. And this is another place where I would encourage families to provide feedback, because we want to be working with our team to make sure that we design the easiest possible trial for our families to participate in, and allow you all to to join in the process without disrupting your routine as much as possible. So we'll want your feedback in that process as well. And then this is kind of where we get into like the rubber meeting the road, and where we are going to be asking for some input. And essentially we will be reaching out to the advocates in the space for conversations with the families around a couple of points of engagement. And Right now, the most pressing will be on formulation, on protocol feedback, which is I think what Luke is going to speak to a little bit, and regulatory interactions. So sort of separating those out to help all of you understand the places where you might be able to be interested in providing feedback, and we can maximize our time talking. So Luke, do you want to tell us a little bit about the protocol feedback situation?
Yeah, sure. I just have three things to say. One is when I talk about protocol, I think feedback is not a good word, because what's the use of coming to somebody and say, hey, can you give me feedback about this shitty protocol that's already been submitted and is under consideration, right? So it's less about feedback and more about inclusion in the creation of, right? And I think that feedback for other things is good, but when it comes to protocol design and development, feedback is too late, right? It's too late. And it's when you're creating it, if you're creating it together is incredibly important. And there's an example, Hans and I, when we were working on a different rare epilepsy study years ago, not too many years ago, but we decided that we wanted to do whatever we could to completely remove the burden of participation for families who are going to enroll in our study, however we could do it, right? And it became very practical. So Hans and I set out to, let's remove the burden of travel, right? Let's get rid of, it's hard to, have, companies who work with CRO travel agents to set up travel for families, because then those third parties don't understand that you can't have a kid like ours, or community like ours, have a connecting flight in the middle of the night, sitting at the back of an airplane, because our kids, it often, fatigue is a trigger for seizure. We have a wheelchair, a dog, a walker, everything. You need to have these considerations in the front of your mind. And the way to do that is to often eliminate that unnecessary third party for the company, right? So people's default is, oh, that's not compliant. Well, in fact, it's certainly compliant to be able to talk to the community and say, here's how we eliminate the burden of travel and make it easier for you to participate in this study. But, and so we are somewhat successful at that in sort of creating, and that accelerates the entire process, because if you can do that, then more people are going to say, yeah, I want to be in that study, right? We're working with a company who understands that, while this clinical trial site might be a 2-hour flight, once you land the airplane, it's a 3-hour drive to the site. So it ends up really being 5 hours, where if you're at a clinical trial site that might be an hour-long flight, ultimately it's 20 minutes from the airport, so it's less time. So if you understand all of these things going into the clinical operations and development, that's really important. But the example I was going to give is, we had a group of caregivers in the room with us when we were going over we're reviewing the protocol before it was submitted. And at one point, somebody raised their hand, and it was a parent, a mother, an advocate who worked with this one specific community, and she said, that will never work. Like that solution and that formulation won't fit through my daughter's feeding tube. We'll have to rinse it out, wash it out. It won't be able to be compliant because they're not going to get the drug in. That would have ruined everything if you started a clinical trial and didn't understand and figure out that patients weren't getting drug because it wasn't the right, solution to get through a feeding tube. And, just by that mother saying that, everybody in the room and the clinical development folks immediately made that adjustment. And if the clinical trial had already started, we would have had to file an amendment with the regulatory agencies. We would have had to start all over again. Patients who were originally in the study would have been disqualified because they had, experience already in the study, and it would have messed everything up. So just by having that mom raise her hand and say, that would never work with my kid's feeding tube, the study happened faster and more correctly. So it's that kind of engagement early that's critical. I think that was the story.
We had a very similar thing in my community where I timed myself opening capsules of granules, and the amount of effort that goes into opening each one of these capsules and then realizing that no one had figured out how you put the capsules into a syringe and then push it through a feeding tube. I think these are the kinds of questions that parents ask, but you don't think of if you're, in a room, never having dosed a med by feeding tube before. So I think it's, I always, when Jillian came on and she said, I'm not a doctor, I'm just a mom. This is exactly, this is exactly who we need to hear from, our moms, and obviously doctors too, but I think.
And don't forget Keith and I, dads too, here.
Yes, absolutely. All the dads. So we will have an opportunity to be discussing formulation questions with the community coming up very soon. And to Luke's point is we are looking at co-creating the protocol. I think we have some opportunities for feedback on study design coming up soon. And we'll also be having discussions about regulatory interactions. And to that point, I put together some examples that I went through with the 8A community, just to give you a sense of, in my role as the director of the Acute Syndrome Foundation in the SCN8A community, we had several points of interaction with FDA that were really important to moving the study that is now enrolling in our community forward. The first point was there was a slowdown in our study, and I did some outreach to FDA in November of 2020. And essentially what I did is I wrote to them, and I wrote the letter I'd always wanted to write to FDA. I don't know why it took me so long to do so, but essentially said, I'm exhausted and I'm tired of watching things go slowly, and here's why. And I told stories about a few of the children in our community. We had just lost 2 children in a matter of a week and a half. We had another child in the ICU. And I just told FDA in 3 or 4 pages what that was like and what that meant and what our community was doing in response to these challenges. And I expected it to be read by somebody. I didn't really realize that it would get the reaction that it got. And what wound up happening in this case is that within 24 hours of sending it on a weekend, I had an email back from Janet Woodcock, and then I had an email the next day from Billy Dunn, and they asked for a meeting. And so 3 days after sending this letter, which I was a little afraid that it was a bit too impassioned, 3 days later, I had a meeting with Billy Dunn, among others, and basically to talk through the needs of our community, what the unmet needs were. And in preparation for that meeting, I had about 24 hours. And what I did, and Liz and Keith, I'm sure this sounds familiar to you, but what I did is I posed a question to my community in our Facebook group. And I basically said, I'm talking to FDA tomorrow. Tell me what you want them to know. Tell me what it would mean to have a precision treatment sooner. Tell me what it would mean to, what concerns a targeted treatment would alleviate. Talk to me about the risks that you're already taking in your day-to-day medication choices for your children. And within it was 12 hours, I guess, I had 40 responses from families, and I went through quickly and kind of themed them. And so I was able on that call to talk through the large themes that I was seeing. And namely the biggest one being that our, FDA's job is safety, and that our children were not safe at baseline. They're not safe right now. And so we need to move forward faster in order to address that. And the result of that meeting was an opportunity to send an encouragement from FDA to send them a packet of all of those responses. So I gave the families another 24 hours. I collected the responses of people saying things like, I have an 11-year-old who's addicted to benzos. That's my baseline. I I'm considering taking my child off of their anticonvulsants because they're seizing anyway, and then we have all of these side effects. And I think it was a really powerful set of documents and set of words. And that was the community. That was our community saying, you know, I'm going to raise my hand and I'm going to say what my concerns are and what my fears are. And then additionally, those of you who were on last week know that we had Rachel Callender in on our meeting last week, but she also had come and worked with the SCN8A community to develop these awesomeness forms. And I also sent those into FDA because I felt like, you know, we can talk for ages and ages about what drugs our kids are on and what their side effects are and what their quality of life is, but they're children first. And this is what I love about Rachel's awesomeness forms, is that it really paints a picture of who our children are and orients us toward what we're fighting for, which is, yes, fewer seizures or better cognition, better communication, but really it's a better, it's more opportunities for our children to be what's beautiful about them already.
Yeah, that's awesome. I also think that all of what it's incredible, Hillary, by the way, but I also think, and how it relates directly to R&D is, you know, it helps make choices that companies are making with endpoints and measuring, you know, less arbitrary, right? So you don't have a room of people developing a clinical trial sitting around saying, oh, I don't know, let's make it 15% seizure reduction. You know, you actually have a clear sort of a way to understand what really is meaningful. Like for our family, you know, it would be awesome to see Susanna stand up and run a marathon, right? But 5 steps would be a game changer. And if there was an intervention that could give us 5 more steps, like, sign us up, right? And all of this that you're doing here is remarkable because it makes things a little bit more specific and less arbitrary for companies developing clinical trials, I think, and regulatory agencies.
Yes.
Yeah, it's, well, by the way, you heard names, I don't know if you're familiar, but Billy Dunn is the person that eventually tell us if we can go on with the next study or eventually the drug is approved or not. So it's pretty influential in what we do. And I want to, you know, reconnect to what Luke was saying. I mean, I know that we debated with the group, you know, of course, for us, it's easier to measure seizures, but we also know that that's not the only problem. And actually, it's one of the several problems. Some kids, they don't even have a seizure, but they still have problems. So in spite of the challenges, I think we might manage to include, you know, the kids without seizures, if we can understand what we can say to the FDA, say, look, this is problematic. We have probably no perfect way to measure it, but we can measure to some extent. They will help collaborate. This is why, you know, in these circumstances, it's so important that if we do that together, we can achieve it. You know, we can increase our chance of success. That's what I was saying.
Yeah, I think when you think about it as aligning as parents and caregivers, what that meaningful change is, and as regulators, what they can measure and understand, and us as a company sort of acting to help facilitate those discussions so that we're able to, as Hans said, you can have a good drug fail because you're measuring the wrong thing. And you know, we have to work together to figure out in this community what the right thing is to measure and how.
No, the hardest thing, I think, and you know, Pierre, Andrea, and Hans and Hillary, you guys might, I don't have the answer to this, but you know, especially with non-seizure consequences of these, you know, the diseases that we're all living with, is just as important as making it, you know, measurable and quantitative and accessible is making them standard, right? You have to standardize them before they're actually, you know, going to be. So what can we do as a community to make these measures standardized, right? So when we do get to FDA, the company isn't coming with this new concept, but the community has already done the legwork to somewhat standardize this notion of, you know, this would be a meaningful change. And it's not a new concept for a company to come and say, hey, can we do a can we use the Xeno walkway to measure gait? And could that be an endpoint, right? Because the community has already contacted, just like you did, Hillary, contacted the agency and other stakeholders who are ultimately going to make the decision and say, yeah, that's an appropriate endpoint, because it's sort of, we're hearing a standardized sort of response from the community that this would be meaningful.
Yeah, I actually think about this a lot with communication as a measure. My daughter, who's nonverbal, has had probably 18 months of the most rapid advancement of her communication skills that anyone could possibly ever have predicted or imagined. She went from putting 2-word phrases together with her eye gaze device to writing full fiction stories, multi-character fiction stories in a year and a half. But if the tool with which you measure that change of communication, it doesn't account for an adaptive communication device, you can't move that measure. You can't prove that there's any difference. And so that's, again, to Luke's point, that if the community has already sort of had accepted measures, accepted notions of how what is meaningful, and communicated those to regulators, we are able to sort of pick up from there.
Can I ask Keith and Liz and the other leaders about, have you guys sort of galvanized and been interacting with FDA and people on the Hill and otherwise?
We haven't a lot. No, I don't know about you, Liz.
No, we haven't either.
Awesome. It's good next step. I'll join you.
Can I ask a silly question? We're talking about the FDA now, and of course that's great. Don't get me wrong, but there is also the EMA, the European Medicine Agency. How does it work once we have an FDA approval? I think we're back to square one for Europe, and we have to replicate everything all over again. Is there perhaps a possibility to, as the companies did for COVID, do both procedures simultaneously, so with the EMA and with the FDA, so that access to radioprodil or whatever drug or cure could be earlier available for Europe as well?
Yeah, Gillian, I hear you, and I think maybe we are the only two on this call that we are on this side of the
From Europe.
But I can assure you that we doing our best to progress in parallel the program, in both in Europe and the US. So the intention is to open sites in Europe and US, and then try to have submission with the EMA. The EMA, I would say, as everything else we do in Europe, is a bit more complex, because it's made of many countries with many different languages, and also traditionally they have less maybe experience than the FDA, but nevertheless they're also very open to orphan disease development. They also have an opportunity to interact and to hear from us. Your point is well taken, and although we have a clear precedent with the FDA, now I ask a question to Hillary if they did anything similar with EMA.
Yeah, we did actually. We took a slightly different approach, and that was we waited. Well, we had a very different sponsor on this program, so I didn't have the visibility onto the timeline that I think we will be. I know we will be providing to the community. So I didn't interact with EMA until I was specifically told that there was a good opportunity to do so. But I took a slightly different approach, which was to handpick stories from families in the countries that were of question that for sites. And we collected those stories, asked similar questions, but it was more tell the story of your child and your child's condition and what the implications are to your day-to-day life. And we pulled those together in a packet with the child's name, their age, and the country that they lived in. And I did take a few that were not in countries that we would have sites in, because they were particularly powerful stories. And I felt like it was enough that they were European. But I think it was very helpful to have, 6, 7 stories from Italy, so that we had, we knew that that was being seen as, this is the Italian population, this is what they're dealing with, so that it could be read sort of in a culturally significant way as well. And that was the largest population that we had duplicate letters from. So it really starts to look like, which advocates are most organized in which countries, and trying to pull those together. But my recommendation would be that advocates be collecting those prospectively, so that you would have, a packet of those types of stories that are looking at the kinds of questions that regulators would take into consideration. So impact on quality of life, current treatments, questions like that.
I also think it's completely different so regulatory engagement from the community and FDA, and regulatory and community engagement for regulators in the EU is really different. It's a different set of expectations and really guidelines, right? So I think that when Jillian, do you know who Eurordis is? Maybe Hillary can connect you with the folks at Eurordis, because Eurordis is kind of it's the European NORD, essentially. They work on policy, but they are also very responsible for, they actually have a training session where, you connect with the community, connects with Eurordis, and then Eurordis creates a plan with you, how you can engage with European regulators. And it's a much more efficient sort of galvanized approach, but you have to connect with Eurordis, and his name is Jan Lacam. I'm sure Hillary can introduce you to Jan.
Okay, I think Sandra and Christian might have some contacts there.
Yeah.
Hillary, you've been in contact with them as well.
Yes, yeah,
Awesome.
I believe that they have been working with Eurordis, but we can certainly plan to have a separate focused on European regulators meeting, so that we can be sure that we're giving appropriate advice.
And Jan's a real
it's
straight
very
shooter.
neat
Like,
that they have a head up. Sorry.
no, go. I was just gonna say, you can say to him, Jan, we want to make sure that our community can help this company, or any company, or whoever's making medicines for this disease, get approval faster. Can you help us come up with a strategy of how to do that? He's very,
At the very least, if they have a heads up and know that it's something to take action on, and not something that's going to take another 5 years. Because basically, when I look at my daughter, for example, she's turning 14 in February next, and I'm already kind of giving up hope for her. And our children are aging constantly, and the sooner we have a cure, the better it is.
We hear you, we understand, absolutely. But this is like a very important point to give an emphasis on. As I said, that's our intention to progress in parallel as much as we can. We will do that in Europe as well.
Yeah, and this, as we go forward, we're trying to figure out how you bring the medicine for everybody, no matter where they are on the planet, right? So we're having similar thoughts and conversations about, Pierre-Andrea talked about the compromise and how you move things forward. How and when do you bring in Asian regulatory authorities as well, and just the rest of the globe? So we've spoken specifically about the FDA and the EMA, but we're having similar thoughts and discussions around other geographies as well.
Definitely.
As a Canadian, I'll just mention Health Canada at some point.
It's a good point. It's a good point, though. It's a good point, Keith.
And the
Canada's a
timeline
hard egg to crack. Yeah.
It is very challenging.
We actually have just to say to Keith, Canada is not forgotten. We actually had our first meeting of part of the board of the company in Montreal because we couldn't go to the US. So in Canada, in Montreal site, as it's Toronto as well for sick kids as well. But there's clearly a lot of expertise we want to tap into. There actually was a recommendation from our chairman that was there.
Oh, good. And I think the process is more similar to the FDA than a lot of other countries. So
I do want to be mindful of our time. I know we're having, I felt pretty certain that this conversation would be so interesting that it would roll roll over the, kind of break the seams open a little bit. So, but I want to be sure, Keith and Liz, if you guys have last, some questions that you would like to ask. I know you had a big long list and we're down to the wire, but perhaps you guys want to put some questions to us, maybe one each, and then we can wrap.
Sure.
Go ahead, Liz.
Okay. My questions were really specific to what the trial is going to look like. And so I'm thinking maybe you guys won't have the answers to these yet. And this would be more of a conversation to have next year, but I'll just shoot one of them out anyways. So when we're talking about trial sites, I'm assuming whoever's enrolled is going to have to go in person to a specific site, and you'll have multiple sites, sounds like, in the US and in Europe.
Yes. We haven't yet decided which one they're going to be, but yes, we need to select the first number of sites.
Okay.
Where there will be expertise of the people, clinicians that know how to deal with the kids, and second, there will be kids that can be enrolled. So they will have to understand, to Luke's point, if someone is traveling so far away, how we can also optimize that. We'll do a first visit in one site and maybe follow-up visit in the nurse locally, so they don't have to do the traveling that is sometimes it's unbearable and put stress that it might not be necessary. So how we can optimize that? It's still
Liz, you know what KIF1A.org has done, and Keith, I don't know if you guys have done this too, but I think it makes it helpful in de-risking that for any company who's making medicines, is put together a heat map of where your families are. And so you have a heat map of where your families are, and then on top of that, put where the clinics that they might go to are, and then give it to Hillary and say, look, if we had an ideal list of clinical trial sites, this would be it. I think that's a
terrific help to look. I mean, you come up with one of the most useful things for sure. Exactly, we have to, go on.
We did that in 8A with Drive Time, ordered a mapping software, and we put all of our people, and we used drive times because we felt like in our community, people were more likely to want to drive 3 hours than they were going to want to fly 1 hour. And so that was a super helpful map as well, because that was another instance where we were able to provide feedback, because there were a bunch of sites going in places where there were just no patients.
Yeah, but and Keith, I'll use sick kids as an example. Like, you could pretty much de-risk any enrollment. Enrollment should be so easy for orgs that are so active, like Liz and Keith and yours and ours and everything, because you can literally say, look, we have 15 families in the greater Toronto area. We could enroll 12 of them at sick kids. This should be a clinical trial site, right? So I think that you guys can really play a part in accelerating that process for any company by saying, look, I'm going to do the operations for you. You don't have to do any diligence. Here's how many kids can enroll in this clinic. That's a huge tool.
Yeah, and I think one of the challenges, and this sort of leads into my question, but one of the challenges is that in this study, we're going to be looking at a very specific subset of the broader population. So, it's GRIN 2B patients with gain of function, and possibly also people with with refractory seizures that haven't been treated. And like I looked on the GRIN portal, and like it only lists 16 patients. This is the Banky Lemke study. Only lists 16 patients that are gain of function of GRIN 2B and have or had seizures. So presumably there's a smaller percentage of those where seizures haven't been treated. So it looks like the known universe, at least of people that are in the patient registries to date, is quite small. So maybe that's part of the reason why you say that you're willing to look at endpoints other than just epilepsy. But one of my question around that is that I believe that, to really ensure success, if you're looking at endpoints like cognition or speech or vision, that typically, you would want to make sure that the patients are getting physical therapy or speech therapy or vision therapy at the same time to really bolster the outcomes. And if you're doing that, then the trial probably needs to be placebo controlled. So do you anticipate doing all of that for the larger later phase?
No,
Or is
I
it
think
too early?
Keith, yeah, thank you. This is a critical point. We actually look at with Johannes last Friday at the portal, and look at probable or likely, let's say maybe gain of function clearly established or likely to be gain of function that we might want to recharacterize with Stephen. I think the number is 75 subjects on the GRIN to be. So it's half of where they are under 60. Not all of them are seizures, but we're really open on not having only seizure patients at this stage.
Okay.
Because again, we knew it, but hearing from you again and again, we say, gee, we can't go that way without ignoring that issue, right? Just now we haven't closed the book on that because, but even though there are those challenges on the endpoint you name, we're still going to start with the open label, with no placebo, because we need to learn how those endpoints are going to perform in this initial stage. Are these the right one? We can have a more liberty in this first phase to choose a few, and then say, okay, now we're going to pick the winner for the next phase. And then we're going to go randomized versus placebo for a little stage, and then, move on. That's kind of in a rough, plan of everything we're planning to.
Okay.
And Keith, from a heat map perspective for dividing the community and looking at the broader community as well as the more narrow community, I think it would be really helpful for you to develop a heat map with those labels, so that you would as a resource going forward for other people in the space as well, that you would be able to kind of pull on and off various subgroups. And I did that with our heat map with just with age groups, because I knew in a particular instance that we were looking at 2 sets of age groups. So I was able to turn those on and off, but certainly you could do it with the subgroups as well.
Yeah, I think especially for US families, Liz probably has better data than we do, but we can work together with Liz and GRIN to be Europe to come up with that.
Yeah, and you get really.
I cannot forget. No, sorry, look, because this is you can't imagine the value of this for us. It's such an incredible. I think the plan was, as we started with Johannes, to go to those 75 and see where they are located, and we're going to do that nevertheless. But as you know, this is only half at best of the population that we are aware of. So yeah. So
this is great to be doing in parallel with if you guys are talking to Hillary and the team as they're developing their criteria, you could get really specific with your heat map and your genotype, phenotype, whatever, and just say, okay, just let us know what criteria you're looking for. And literally, here's 15 kids in this location that go to this clinic that meet those criteria. I think that you can de-risk it that much, just by that legwork. And then in doing so, for the companies that are developing medicines, you're also strengthening your community, right? Because you're understanding more about every kid who's living in every different part of the world. So it's a great example of developing tools in parallel that are going to accelerate the development of therapeutics and also strengthen a community. I think that's a huge point. Yeah.
Just out of curiosity, Luke, if you were doing that for KIF1A, would you do that with Wendy Chung's registry, or do you have like a separate registry of just contact registry that you guys
We've done it, and I can show it to you, and you can steal the whole process. And so we've done it with our registry is in parallel with Wendy Chung's, right? So we have a mechanism in place when a family reaches out, they become part of our database, but also go directly to Wendy's natural history study and registry. But we've actually mapped out clinics and populations and where they are, and I can walk you through it at any time, Keith, and anybody who wants to steal that process, it's all yours.
Why don't we add to our list of meetings that we're going to have coming up, and we can do and this is for everybody who's listening, we're going to be interacting next 3 weeks in a row, but on Mondays again, but an hour later with GRIN 2B Foundation and Cure GRIN and hopefully GRIN 2B Europe to walk through a couple of very specific topics in detail. And I think we can add the heat map to that. I can show my process as well, which is a little bit different because we don't have a parallel natural history study and foundation registry. We have just like a contact registry with some basic information. So we have variants and date of birth and a few more pieces of information as well as location. And then we used a mapping software to kind of plot that all out. And it was quite simple. So having a couple of different options or looks at what it could be like might be helpful. So Luke, I'm going to bug you again.
No, yeah, absolutely, absolutely. And whatever we've done is please steal away and replicate,
Yes.
make it better, and then we'll copy from you.
At this point in time, are we also allowed to speak to doctors? Because I know in the Utrecht Hospital in the Netherlands, there's been a program for kids on memantine, so that's definitely a gain of function. And I know it was mainly GRIN 2B children. And I think I may have a better chance of convincing families by going through that specific doctor who run that specific project than going straight to the families. Is that also a possibility, or is that something to put on hold for the time being?
I think it would be great if you would actually put us in touch with that doctor and we could meet with him or her and get a sense of what the population is and then do outreach through. Is that what you were going to say, Pierre Andrea?
Yeah, no, absolutely. And I think it's, well, we also have a good part of the team is Dutch speaking, being Flemish. And there is a conference in the Netherlands that is going to be attended by our clinical operation colleague that is Dutch speaking. It might be an opportunity actually to meet with some of the doctors.
Well, there are a couple of people you need to meet here in the Netherlands.
Yes, but irrespective of the conference, feel free to give our contacts to any of the colleagues out there in the field that we are happy to interact, and we want to do it.
And then you're referring to the conference on 21st of November in the Erasmus?
Yes, I think that's the one. Is it Rotterdam?
That's not the place to be.
No? Okay.
No, it's not. It's not. But that's a terribly long story. But the Erasmus
Gillian,
actually.
I suggest that, because, we can, on our regional thing, can you drop me an email? I'll put you in touch with Mark Bostille. That is, you can tell all, also to me, but, he's the one that's speaking that was going to attend that conference. We want to know all the background.
If you can drop me an email of that person, I'd be happy to fill him or her in.
Okay,
Yes.
we'll circle back and make sure that oh, wonderful. All right, I know we've gone plenty over time, which is
No problem.
wonderful because we have tons of things to talk about. But I just want to say again, thank you to Luke for coming and sharing his expertise as always.
Thank you for having me. It's so wonderful to be on the call with so many faces that I just am so appreciative of and love.
Yeah.
Thanks,
well,
Luke.
Thank you. Thank you, Liz and Keith, I'm sorry, we ran over and there wasn't too much time for moderating questions, but we really appreciate it. And I just want to encourage everyone who's on to reach out to Liz and or Keith and Denise and let them know if you want to be involved in further, detailed conversations on the topics we talked about, because there will be opportunities coming up in the coming weeks and months to dive into some more intense conversations about the details of things like formulation and protocol and regulatory involvement. So with that, I'll say thank you to everyone for coming and close us out for today.
Thank you so much.
Thank you.
Okay,
Thank you.
Bye.
Bye.
Thanks a lot.
Nice to meet you, Pierre.
Bye.
Thanks, everybody. Bye.